I'd like to thank you all for joining us tonight. We're going to be talking about bovine tuberculosis, which I like to call the bacterial ninja for reasons that should hopefully become clear to all of you on this webinar. With me tonight, I am pleased to have ABP's Alberta Beef Producers General Manager, Brad Dubois. He will be speaking a little bit later in the webinar, as well as Dr. Noel Ritson-Bennett from the Canadian Food Inspection Agency, who is the incident commander for this current tuberculosis investigation stemming back to a case in 2024. The other piece that I want you to be aware of is that we will not be answering questions about individual circumstances. If you have particular questions about something that happened on your operation during that investigation, we will not be answering those live, but we will potentially follow up with you offline. So this webinar is really to explain about what bovine tuberculosis is, why we care about it, why it's so challenging, why I call it the ninja. And we will also be going through some frequently asked questions that I get a lot. My name is Karin Schmid. I'm the Beef Production and Extension Lead with Alberta Beef Producers. I'm also serving as the industry liaison in Alberta for this particular investigation. And this is a role I also previously played in 2016. What is bovine tuberculosis? It is caused by a bacteria called Mycobacterium bovis. And there's actually 120 different species of Mycobacterium, at least. There might be more. And they share a high degree of DNA sequence, which is important when we talk about some of our diagnostic tests. It is sensitive to heat and sunlight, but it actually is also fairly resistant to cold, which is something you may not think about. We have no real practical on-farm treatment or vaccination for this disease, and it's transmitted by frequent or extended nose-to-nose contact. Less commonly, it could also be transmitted orally through contaminated feed or something like that, through semen, or from the dam to the fetus. It spreads through the body via the blood or lymphatic system, and it's really a slow, progressive type of disease. It takes a long time to grow and to show any clinical signs. It can also lie dormant or latent. So what it actually does is the body tries to wall off the infection and essentially forms these lesions called granulomas. And I'll show a couple of pictures of that later. So if you're a bit squeamish, you may want to just look away when I get to that point. But essentially, that latent infection can be reactivated at different times. And it also has super shedders. And you may have heard this term with regard to other diseases. E. coli is one that animals can also be super shedders. And we don't really know a lot about what makes an animal a TB super shedder or what doesn't, but we do know that they exist. In addition, different strains have different virulence, and so they may be more infectious than others. And generally, like I mentioned, we don't really see clinical signs until the animals are very far advanced in the disease process. So this is just a bit of a diagram of how the infection causes different disease states or not. So here we've got our little bacteria, and it hits the cow. The cow has an immune response. If it clears the bacteria, they will not be infected. But sometimes they will have an immune response. Again, later when they're exposed again to the bacteria, they might clear it again, which is cool if they do and become non-infected. But other times, and I've shown it with the little lightning bolt, they may manage to, let's call it, suppress the infection. And that's where that latent infection piece comes in. Otherwise, if the bacteria is not detected by the immune system and kind of multiplies and runs rampant, we can hit active disease. But active disease can turn into latency and latent infection, and those can kind of go back and forth. So latent infection may be activated with things like stress, old age, essentially anything, maybe another infection that is impacting the immune system and causes that TB bacteria again to cause active disease. So here's the trigger warning. This is this is some bovine lungs, and you can see in here that granulomatous formation, really that lesion that is very indicative of TB. But the lesions can also look super, super small. So in that circle, we can see that tiny, tiny lesion. So it can be very pronounced or it can be very hidden. And this causes us some problems when we go to think about how we detect and how we manage tuberculosis. But why do we even care about this disease? Well, as we probably all know, 50% of our beef production is exported, which is a significant portion of our market share and supports our beef production systems. The global economic burden of tuberculosis is about $3 billion annually. It is the leading infectious cause of death worldwide, and 25% of those deaths are in children in human tuberculosis. Milk pasteurization has actually been a super good human health story in terms of reducing this disease because it is zoonotic. It can transfer to humans. Historically, anywhere from 10 to 30% of human TB cases were caused by the bovine form of tuberculosis, and milk pasteurization did a lot to decrease that. In Alberta, there was a retrospective study done not that long ago, and it shows that success. So from 1995 to 2021, there were only 20 cases reported of tuberculosis caused by the bovine form of the disease. And none of those were domestically acquired. The last report of a domestic case occurred in 1991 in a veterinarian. In the US, anywhere from 1.3 to 1.6% of the human cases in that time frame referenced there were due to bovine tuberculosis. But in Mexico, and here we see the impact of milk pasteurization, they're running around 30% of human cases. And primarily that is either in workers that have very close contact with infected animals or due to the consumption of raw milk. But even that being said, this was an article that was posted in 2025 where a very young girl who is older now, she's 25, something like that, became infected with bovine tuberculosis, and it put a very big strain on her body. She continues to deal with long-lasting effects. So even though it is a relatively low prevalence and relatively low risk disease in the human population, it can still have very significant individual impacts. We have seen about 100 years of eradication progress for bovine TB in Canada. And I'm just going to go through a very rough timeline here of how that all started. In 1923, when the eradication program really began, we had a prevalence of the disease of about 5%. In 1961, we completed the first national herd test. So that was all animals, all cattle, all susceptible species. It was around 50 million tests. There were 400,000 reactors removed, and 4 more of those national herd tests would be completed by 1980. Through that process of removing those reactors, we decreased the prevalence to about 0.1%. And then in 1978 to 1984, the whole program went under review to see if we were approaching this eradication properly. And what we did at that point in time after the review was really move to a slaughter surveillance model, whereby we look for those granulomas upon slaughter and then move to the epidemiological tracing, whole herd, and all of those investigative pieces where we really trace where the infection may have spread to and may have come from. And that's a system we're still working under today. In 1997, the US finally removed testing requirements on Canadian cattle going to the United States due to our progress with the eradication program. In 2003 to 2019, this represents the Riding Mountain eradication area in Riding Mountain National Park in Manitoba. As many of you probably know, there was a wildlife reservoir implicated in that period of time, whereby any cattle coming out of Manitoba to the United States had to be tested. There were specific programs in place to reduce the contact between the wildlife that were carriers as well as the cattle that likely spread the disease to the wildlife in the area in the first place, and the eventual reduction of the wildlife population in that area. And in 2019, finally, those restrictions on that particular area in Manitoba were removed. In 2020, CFIA added additional testing requirements for cattle coming up from the US that were rodeo stock. And right now, our prevalence is estimated at somewhere between, right around 0.01%, which is very, very low prevalence. And it's actually less than the World Organization for Animal Health standards permit. So over this whole 100 years, we've done over 100 million tests. We've removed 800,000 reactors and paid out over $130 million of compensation to try and get this disease to the state where we have it now in Canada. And thanks to those efforts, all areas have been considered free of bovine tuberculosis since 2006. This is just a bit of a graphical way of demonstrating this. We can see some cases here. Oh, sorry. I need to go back to the previous slide. This is where we move from test and remove to the whole herd depopulation scheme. And that's where you can see we really started to make a dent in the case prevalence. And we can see that our cases have become increasingly rare and sporadic over the past 20 years. It may not necessarily seem like that with 3 cases in the last 3 years, but time will tell whether that's an anomaly or whether we're seeing a resurgence. And personally, I think this is likely just a blip in the terms of our ability to control bovine tuberculosis. So why did we move to that whole herd depopulation, the slaughter surveillance scheme? Well, as part of that review in the 70s and early 80s, a cost-benefit study was done, and the reference is there on the screen, but they found that the return on investment was 21 to 1 for test and remove, but 34 to 1 for depopulation. And a similar study in the United States going through to 2003 shows that they've returned 13 to 55 billion over the cost of the control program to producers. This also protects an industry, our Canadian industry, providing 34 billion to our GDP, and we're top 10 in beef production and beef exports. And so having this tuberculosis response and tuberculosis free status really means that we open up export markets that we may otherwise not be able to achieve. So I want to go through and talk a little bit about the disease response actions and how they can impact individual producers and why we do them. So this is kind of the first step here. So we may detect a case on slaughter. We trace that case back to their home herd, that herd of origin. CFIA starts instituting movement controls. They do the epidemiological and tracing investigation to find out where all the animals came from and where they went to. And then there's the testing component. And the testing component I'll get into in a little bit more detail, but really it also includes that removal of reactors, which is the removal of them generally through slaughter processes and the enhanced postmortem that follows those to look for those lesions. But we can also do a few more things when it comes to the removal of reactors. There are some on-farm options if folks are willing to explore those. Now, if there's a positive, those positive animals also are all depopulated. And we'll talk a little bit more about why they undergo testing before they're depopulated. But then that infected premise undergoes cleaning and disinfection, and then there's a restock testing component. So that's almost separate from the rest of the investigation because it only applies to herds that had an infected animal associated with them. So I want to talk a little bit now about movement controls and how movement controls work in a disease investigation. So let's say, and these are hypotheticals, right? And they won't cover off every situation, but for illustrative purposes, let's say here, this black cow with the X is our infected animal. They have mingled with the brown cows. They have fence line contact with the green cows. They have fence line contact with these blue cows. Over here, there's some gray cows, which are separated by, let's say that's more than what's shown on the screen. It's a significant difference. So they would likely not be implicated in the investigation, except that they purchased black cows. And so now they are implicated because those black cows came from the herd with the positive animal. And I'm hoping to show this in a few more different ways. So bear with me while I do that. But really what we're talking about when they talk about an epidemiological or a tracing investigation is really trying to find out who or what type of animals, when they might have had contact with an infected animal, how long that contact occurred, what type of contact it was. And it gets really, really complicated. This is really trying to get at the risk of disease transmission. And I think this hangs up a lot of producers because the risk of disease transmission isn't always the same. So CFIA will gather a lot of information via what they call a premise investigation questionnaire or a PIQ. And a lot of times that may seem really overwhelming and like, why do they need all this information? But it's really to support this tracing investigation and the epidemiological connection between animals. And I just want to flag throughout all of this that I want to remind people that correlation does not equal causation, and sometimes directionality can be a little bit tricky to figure out. So if we're talking here, I just want to go through the terminology. An infected herd is the current location of an infected animal or the place it most recently resided. So if an animal was found on slaughter, an infected herd would be the place it came from before it was found on slaughter. The lifeline herds are technically a type of trace in herds, but they're different because if an infected herd had an infected animal that came from a different herd and wasn't born on the infected premise, that would be a lifeline herd. So they have a direct relationship to a positive animal in that infected herd. The trace out herd are animals that left the infected herd. A trace in herd are animals that came into the infected herd. And a direct contact herd are animals that commingled with the infected herd. Fence line contact is fairly self-explanatory. And then we also have proximity herds, and these are herds located generally within 5 kilometers of an infected herd, where maybe transmission had happened that we can't catch through the other types of herds on this slide. And all of these herds have different risk levels. And the way the investigation works is generally we try to move from the highest risk level to the lowest risk level. And so we start off obviously with the infected and lifeline herds. Those have a direct relationship with positive animals. There's known exposure. Same with the direct contacts and trace outs. And then your fence lines, your trace in, and your proximity. And to manage resourcing and capacity, right, we want to start where the risk is the highest, and then we move to the lower risk herds as the investigation moves forward. All of these risk levels consider individual herd characteristics and management. So you might think that you're a fence line contact, but there might be something where known exposure could change that risk level if more information is gathered. So I'm going to walk you again through my cows and my fences, and you can see how complicated this could be in a beef production scenario. So here again, we have an infected cow on a premise. They have cows from multiple different sources on this premise. Some of these other cows shown by the red X's are also infected. We have a blue cow in the middle that came from the blue cow herd. Potentially, they brought the infection with them, but not necessarily. In this case, I've got them bringing the infection with them. We also have animals that were sold, right? That were sold from that herd. So this would be a trace out herd over here. Maybe an infected animal was actually sold to this herd, or maybe originated from this herd. And if it originated from this herd, it becomes a lifeline herd instead of a trace out herd. Here we have some black cows again, but this time we're mixing with some green cow. Oh, and this green cow might happen to be positive. And then we have the green cow herd is now involved. Oh, but they also bought cows from the brown herd. And now the brown cows are involved. Oh, but they also maybe bought cows from that original index herd up in the top left corner. So they have black cows. And we're doing this web over and over again with all the animals that originated in that top left corner. Now I've got this gray herd, and I want you to keep an eye on the cow I've just circled there. So that gray cow potentially went to this herd, was sold. That's a trace out animal. All is well and good for now. Maybe it went through an auction market. That's okay. Then it was sold to this herd down in the bottom right corner. And in the process, this herd at some point in time noticed that this gray cow lost her tag. So they put in a new tag. Many people do that all the time, but they didn't actually know what tag she lost. So they weren't able to link her previous tag with the current tag. Now she goes through an auction market and ends up over here. She's been branded twice since then. And so where did this cow actually come from? Because the tag chain has been lost. We don't know, because of the retagging and the lack of records, we don't necessarily know that she came from this herd in the upper right, which traced back fairly easily to the herd in the upper left. And this is sometimes why tracing takes so long and why there's second rounds of tracing, why there might be third rounds of tracing, because we have the auction market system in between there and the brand inspection trying to figure out where this cow came from in the first place. And that's why, and we'll talk about this more a little bit later, that's why records are so very important in a disease investigation like this. Now I'm going to switch gears a little bit and talk about testing. And I want everyone to keep in mind that no diagnostic test in the history of ever is 100%. And our TB tests are maybe not even close to 100%. And so we work with what we've got. The herd risk level can change the testing approach. The live animal tests are either what you may have heard as the caudal fold test or CFT. Sometimes a comparative cervical test is used, that's the CCT, or Bovigam. And those are the live animal tests you'll probably hear referred to the most. So in this particular instance here, we've got this little bump, and that's the caudal fold test. So that would indicate a reactor to the caudal fold test. On the postmortems, so we take those reactors to that initial test, and we do an enhanced postmortem. Well, why would we even do that if we already have a positive on the caudal fold test? Well, as I mentioned way back at the beginning, if you're still with me, there's a lot of species of mycobacteria, and they can cross-react with other species of mycobacteria. So Johne's might be a common one that could cross-react. And so that's why they take the reactors and then do an enhanced postmortem to look for those actual lesions. Once we look at the lesions, they take samples. Whether there's lesions or not, samples will be taken. They're looked at under a microscope slide. So that's the histopathology part. And it's stained. So this is actually some staining that shows the bacteria within a sample that was taken. If that slide looks consistent with mycobacteria, then they may do a DNA PCR to look at whether or not it's actually Mycobacterium bovis or one of the other mycobacteria that could cause a cross reaction. And so if the histo is positive, the PCR is positive, we consider that a positive case definition for bovine tuberculosis. Now, all of those samples, whether or not they show positive histopathology, would still go to culture. And then that is really the gold standard. So we see if we can actually grow the bacteria on a plate in the lab. And if that bacteria grows, there may or may not be another PCR to confirm that it is exactly that bacteria. But that's the only way that an animal can be really determined negative, is if it's negative on culture, because there are certain instances where histo might be negative, but culture will be positive. If anything grows on those plates in the lab, then they'll do a whole genome sequence on those positive cultures. And the whole genome sequence can tell us a lot of information. It can tell us the closest relative for a strain that might have been reported in a different jurisdiction, in wildlife or in cattle, pretty much anywhere in the world. It can potentially give us information on how long that particular strain has been circulating in a particular herd, because they can look at the rate of mutations from different animals and see how long that strain has been circulating. But this is a long process, because remember I said that it's a very slow growing bacteria. So when you culture, it takes a long time to culture that bacteria. So that total testing time is somewhere in the neighborhood of 14 weeks. The whole genome sequencing also, as I said, gives you the strain differentiation. So another thing that we can think about is how can we increase our ability to detect bovine tuberculosis in live animals? So we might do parallel animal testing. So that would be the caudal fold in conjunction with another test, Bovigam or the comparative cervical test. This decreases the false negatives. So we take reactors from either the CFT or the ancillary test. If we take all those reactors, that reduces your false negatives. But it does also increase your false positives. So parallel live animal testing is generally used in higher risk herds. In serial live animal testing, this will decrease your false positives, but increase your false negatives. So in serial live animal testing, you test with the caudal fold test. On the reactors, you do either CCT or Bovigam. And if they were positive on that secondary test, that would be considered a reactor. So what you're doing there on the lower risk herds is hopefully screening out animals that you don't have to get rid of because you're decreasing your false positives, but you do run a little bit of a risk of increasing your false negatives. So I'm just going to run through a couple of flow charts, and I can't go through them all right now, just in the interest of time. But if you take a quick picture of your screen, you can go to either of these links and it walks you through the testing of all the different herd types. So I'm going to talk about lifeline herds first. So again, as we talked about, the first primary live animal test is the caudal fold test. They may, depending on risk level or potentially a subset of animals that may have had more exposure, do some parallel testing with Bovigam. All the reactors, so if they reacted to caudal fold or Bovigam, would go for enhanced postmortem. And then the confirmatory testing would happen with histopath and culture and see whether or not we potentially have a positive animal there. For trace out herds, the process is a little bit different. For trace out herds, every individual trace out animal, they're still tested, but regardless of how they test on the caudal fold test, those individual trace out animals will go for enhanced postmortem and the samples for histopath and culture. The rest of the animals that weren't trace outs, so their herd mates, would undergo the caudal fold test, potentially some ancillary testing. And then again, any of the reactors to either the CFT or ancillary testing would go for enhanced postmortem, and then the samples would be collected for histopathology and culture. If a trace out animal is in a terminal feedlot, that animal is sent to inspected slaughter. And if lesions are found, those samples are collected and then sent for histopath and culture. If a trace out animal is in a non-terminal feedlot, the testing approach really depends on whether the trace outs had any direct or indirect contact with potential breeding animals. So that approach will change a little in a non-terminal feedlot. And finally, I want to talk a little bit about trace-in herds. And trace-in herds are different because the quarantine is usually applied when testing starts. So with trace-out herds, the quarantine is applied when the trace-out animals are identified. With trace-in herds, the quarantine is applied when testing starts. So again, we'll do the caudal fold test on all animals in that trace-in herd. Reactors will be tested again with an ancillary test, either Bovigam or the comparative cervical test. And that comparative cervical test screens out essentially one of the mycobacteria that often cause a cross reaction. And then those reactors only will go to enhanced postmortem with the samples collected for histopathin culture. So as you can see, and again, there's more examples on those links I showed earlier, there are different approaches depending on what type of herd and how you're related to the investigation. In a trace-in test, those non-reactors undergo no further action. So everything is just as normal on those animals. And I want to talk a little bit about early release from quarantine. Anyone who's been part of this investigation probably understands early or conditional release from quarantine. This is actually a relatively new process. It was instituted during the 2016 investigation, looking at those negative histopathology results because we had not seen animals that had negative histopathology with positive culture. Whether or not we can continue to do that is it's really not automatic, right? We were working on data that we have at the time, and with more data, that process may not be there, but it's just important to know that this process is not automatic. It does allow us to resume business early. Obviously, you're asked to keep very good records so that we can trace any animals in case a positive were to occur, but recognizing that negative histopath generally means fairly low risk of a positive culture, not zero risk, but relatively low risk. We felt, and CFIA feels, that this is an appropriate procedure for our industry. The next thing I'm going to do is go through a lot of questions that I hear from producers as they're involved in an investigation. And I hope that this helps talk about some of the myths that might be out there and some of the things producers can do on their end to minimize the pain involved in any of these investigations. So my first question is always, how can I reduce the impact to my operation is involved? And really the answer is records. The answer is records, and the answer is records. I'm not talking about I sent 200 cattle over here 3 years ago. I'm talking individual animal records. The more detailed your records are, the easier the trace is, and there is a possibility that you may be able to exclude animals from an investigation. And so there's often a lot of concern from neighbors or people just in the industry. Well, I'm not impacted by this right now, but what else could I do in the interim, right? And I think it's really about thinking again about records. I would encourage people to update their premise identification number, make sure their contact information is current in the PID system, make sure your information is current with CCIA and your CTLS account, and think about those standard disease prevention protocols. Discuss them with your vet. Think about things like commingling of animals, community pastures. What are your herd introductions or your buying practices? What are your wildlife interactions? What are your colostrum sources? Things like that that you can discuss with your vet and think about small changes you can make to hopefully decrease your risk of bringing something into your herd, whether it be TB, whether it be scours, whether it be BRD or BVD, any of those types of diseases. All of those disease prevention procedures help with those diseases, as well as something like TB. And then stay informed. We've put out quite a lot of information, and I've heard from a couple of different sources, well, oh yeah, I saw that, but it didn't matter to me at the time. So they say they didn't read the article, they didn't read the in-depth information we'd be putting out about the investigation because they didn't think it applied to them at that point in time. ABP Daily is an excellent source of information. The CFIA website, call the ABP office. We can help you stay informed. So again, where can I find help? Reach out. Don't be scared. Everyone has to keep confidentiality at the top of mind. We're not sharing your information with any other producers or auction markets or anyone like that. It's all held very close to the chest. If you have people you trust in your commodity organizations, whether that be ABP or ACFA or Western Stock Growers, reach out to them for help if you're involved and you have questions, if you have concerns. Your case officer, if you're involved in the investigation, can help with that as well, as can the Chief Provincial Veterinarian's office. And don't forget that this is a really stressful time. It's really hard to not necessarily know what's around the corner for your operation, for your livelihood, and make sure you're taking care of yourself during any of these stressful situations. It is not easy for anyone. Can your private veterinarian be involved? Yes, you can actually request to CFIA if you're involved to include other people in your circle to attend meetings with you, whether that be someone from your industry organization, whether that be your private veterinarian, as well as some lower risk herds. They may actually be able to have their testing performed by a private veterinarian that is accredited for TB testing by the CFIA. So they can participate. You don't have to lock them out of the process. The other big question I get is, how do new cases affect Canada's trade status? Essentially, each province is assessed annually through the granuloma. Oh, my tongue is tied tonight. The granuloma submission program, which is the slaughter surveillance, live animal testing numbers if through that granuloma submission program, and a rigor of investigation if a positive case is determined. So we really need to show to our trading partners that when a positive case comes up, we chase down all the leads we possibly can. If we hit a dead end, we hit a dead end, but we really need to show that thorough investigation to maintain that confidence from our trading partners. And there's no automatic downgrade of status unless the epidemiological evidence suggests that we've had undetected spread. So even currently right now, with the case in 2023, the case in 2024, and the case in 2025 in Manitoba, right now, no province has had their status downgraded. Are cats and dogs and horses destroyed on infected premises? Not typically. So usually these animals are all very, very low risk. If there was some specific evidence to lead CFIA to believe that they might be infected, that might be another question. But to date, that is not a standard thing that is done. Why is the trace period generally 5 years? So part of that is that TB, as I mentioned, is a really slow growing, so it has a long incubation period. Our surveillance data from the slaughter plants indicates that there is likely, by the time an animal shows up positive at the packing plant, the disease has been circulating within the herd, not necessarily within that animal, but within the herd for at least 5 years. And there is the ability to adjust the trace period if the data warrants. That's usually done for trace in, as has been done for this 2024 investigation. There's more information on that adjustment of trace in period on ABP Daily. But essentially, if more information warrants, we can adjust those tracing periods. And that has been done in previous investigations as well. Why is my neighbor, friend, person I heard about through the grapevine undergoing different procedures than me? Usually the easiest answer here is that their herd risk level might be different. They might have different individual circumstances that are different from yours. And probably the biggest one is the epidemiological investigation that PIQ shows some different things and maybe put you in a different category. So just because you're undergoing one set of procedures does not mean that your neighbor should be undergoing the same. Why do we bother testing infected herds? Why don't we just depopulate them and have done? Why bother testing those infected herds? Well, one thing it gives us is an indication of the spread within the herd. It allows us to potentially prioritize the epidemiology and tracing, and then it lets that whole genome sequencing be done on all positive animals found within the herd, which can provide us with additional information. So why don't we test and remove instead? We talked about how in the 60s there was a test and remove program. Why don't we go back to that? And one of the reasons is recurrence rates. So in countries that routinely perform test and remove instead of whole herd depopulations, they have higher recurrence rates of TB in the herd. So I'm going to point to a couple of examples. One is France, which does not have a known wildlife reservoir. They do a test and remove policy, and their recurrence rate is 8.3 to 12.4%. So that again speaks to just how sneaky those TB bacteria are and the trouble we have in ensuring that we can identify all the animals properly and make sure we track down all the spread. This is a bit older data, but in the United States in the 1990s, they did an analysis of all of the herds that were under a test and remove policy at that time. And their recurrence rates in those herds ranged from 33 to 50%. 11 out of the 24 herds examined were found to be reinfected, well, still infected, I guess would be the proper term, still infected 2 or more years after they were released from quarantine. And the other piece with test and remove is it generally leads to long quarantines. And we see this in the United States. So I often get asked, why does the US do this differently than us? And quite honestly, they don't do a lot differently than us. And in some cases, we actually have a bit more latitude and flexibility than the US does. Their first choice is still whole herd depopulation. But their indemnity, so their compensation for animals ordered destroyed, is tied to a budget line item. Sometimes they run out of money, and it's a bit of a irritating and long, drawn-out process to get more. And secondly, some of their high-value dairy herds are very against the whole herd depopulation. So they actually give producers a choice. And so if a producer chooses to do a test and remove, they're subject to 8 full herd tests where they are under quarantine for a minimum of 4 years. And then they're subject to 2 annual herd tests again after they're released from quarantine. So that is a really, really long time for a beef herd to be under quarantine. And I would suggest it is not acceptable or even tenable for a beef herd to remain under quarantine for 4 years. So, okay, what happens if trace out animals can't be found? So I'm a trace out herd. I am supposed to have 10 trace out animals on my place, but we did our whole herd test and we only found 8 of them. Maybe they died in the bush. Maybe I sold them and didn't write it down. What happens if we can't reconcile that? Generally speaking, you'll be looking at a herd retest in 6 to 12 months. This one is interesting. Um, because we hear a lot about, well, where could this source be? Where could that source be? And if trace in animals are a potential source, why are they considered lower risk? And the best way I heard it explained to me was trace in animals have the same status as every other animal in Canada. We don't know that there was exposure. We don't know that they brought that disease in. Um, so that's why they are handled kind of at a lower priority because there is no evidence of infection. Now, again, I don't want people to conflate the lifeline herds where a positive animal had a direct connection and just regular trace-in herds. Why haven't we found any sources? I hate that we haven't found any sources, but it's hard. It's really, really hard. Was the strain present but just not detected before? Was a source animal introduced outside of a tracing window or removed from the herd before it could show any signs? Was there a traceability breakdown? We've talked here already about how traceability can break down in the system. Was there a breakdown during import? We know our tests have limitations. What is there maybe an unknown or even uninvestigated transmission route? But it has to be the wildlife, right? Everyone wants to zero in on those wildlife and maybe without precedent, right? Because there was Riding Mountain. We know we have a reservoir in Wood Buffalo National Park. Um, but wildlife surveillance is a key component of every investigation. And in recent memory, the last since 2016 anyway, we haven't been able to tie anything outside of Riding Mountain to wildlife in the area. And let's remember that spillover can happen in either direction. So it can happen to wildlife into cattle or from cattle into wildlife. Um, the strains that we've had in the recent years have not matched any wildlife strains in North America. So is this an unknown reservoir? All of the strains in the last few years haven't been closely related, so it's less likely there's a common source between them. That doesn't mean we don't continue to investigate them, but it just doesn't mean our fingers should be pointing directly at wildlife first. It does leave everyone with unanswered questions, and I get that, but it might just have to be an unanswered question that we live with. Why does CFIA need so much information? So they want to understand herd dynamics, management, potential transmission. The more information you can provide them, the better. Um, I've heard, well, why did they ask about this? No one would ever do this. No producer would ever do this. Well, no one thought people would use their hair dryers in the bathtub either. And that's why our hair dryers now have warnings on them. Noel, I am going to call on you a little bit to expand on why the PIQ is so extensive, if that's all right.
Yeah. As Karen said, I think the PIQ is very broad and quite expansive in its questions, but it does provide a lot of information and allows us to understand management practices on farm, to identify potentially groups of animals that are higher risk and need a different testing protocol, or potentially even animals that are not that don't have exposure or potential exposure to a positive animal or positive herd, and so it really allows us to tailor the investigation and tailor the testing protocols that are used on farm, and it it's very tedious and it takes a lot of work, and a lot of the questions that may seem somewhat trivial at times, as Karen said, sometimes they do result in providing some information that is helpful to us.
So, Noel, can you also tell me why different herds are quarantined at different times? And we went through this a little bit with like the difference between trace outs getting quarantined upon identification and trace ins getting quarantined when you show up to test. But what is the rationale behind that?
And so I think, as you alluded to and you have written on the slide, the whole program, really, the policy is written to with risk in mind and to allow the movement or the continued operation of producers if possible or feasible. And by doing so, it creates a lot of or what seems like inconsistency in our response. Unfortunately, if we were to be consistent entirely across the board, that would mean that everything was quarantined upon identification. So trace in herd, trace out herd, lifeline herd, proximity herd, whatever you call it, we would, if we wanted to be 100% consistent, we could just quarantine everything and then wait till we tested and had final culture results. Obviously, that's not really palatable. It doesn't work very well for industry. And I think what the policy attempts to do is to try to take that risk of disease spread that exists and sort of take into consideration of the impacts that we have on industry. And certainly we recognize that any involvement in the investigation has a tremendous impact. But we certainly try to minimize that.
You kind of preempted my next question, so I pulled it up while you were talking.
Oh, sorry, I didn't even notice.
Was about the consistency, right? But I think we covered that. And I think the point being is you're trying to follow the science, and you're trying to potentially make the least impact while still ensuring that disease control procedures are followed. Would that be fair?
Yeah, I think so. That's fair.
Do you have to reach specific testing numbers in an investigation?
No. So there's nothing that says you must. There's a specific number of animals that are tested, but I think there's an expectation that we do a thorough investigation, and that we're able to tell our trading partners that we've completed a thorough investigation, that we've looked, as hard as is reasonably possible, and that that we're that they trust that we've done that investigation and done it a complete job.
Perfect. Noel, can you give us a bit of a rundown of where we are in the current investigation? And by current, I mean the 2024 investigation. So this is the one that was identified as slaughter in late November 2024. It spent about 3.5 months at a feedlot in Alberta. That was eventually traced back to Saskatchewan. The strain was never previously seen before in North America. I will just quickly mention before Noel gets into some numbers that the dairy case that was identified in June of 2025 in Manitoba is an unrelated strain, but there has been some crossover with some feedlots with trace out animals involved in both investigations, as you might expect with the way our industry works. But if you would just maybe walk us through what you know as of last Thursday.
Are you gonna?
Yeah,
you're gonna push buttons. So
I will.
So again, I think the 2024 investigation to date has identified one infected herd. Within that herd, there were 27 additional animals that were found to be positive for tuberculosis. Total, I'm not sure what the total 154 is, Karin, actually. Sorry.
Maybe I should do this instead. Noel and I maybe should have practiced better. This is the number of herds that have been touched, I will say, in some way by the investigation, contacted potentially, whether that be trace in, trace out, lifeline, et cetera, et cetera. Most of those have been actually not needing any follow-up. They were PIQs were done, the animals were dead, there's no further investigation needed on the bulk of those. So we've had 7 lifeline herds, including that feedlot. All are currently under full or early quarantine release. That's right, to your knowledge. Okay. Trace out herds. We've had 122 trace out herds involved in some way. Again, like I said, the bulk of those have been, no further investigation was required on the bulk of those herds. You can see how many have been or are currently under quarantine and how many remain for the epi investigation and PIQ to be completed. And our trace-in herds, currently that's 11. We do expect this to grow. 7 remain under active investigation. And we expect this to grow due to the expansion of the trace-in window, which, as I mentioned previous, is not uncommon in these investigations. Again, we have 1 direct contact herd. This may or may not grow based on new information. And right now, proximity herds, so that's within the 5K, is about 13. And that number may grow or may not grow, Noel.
I think we're still working our way trying to understand proximity herds, so certainly that number could increase.
Perfect. I just want to switch back a little bit and talk a little bit about compensation. CFIA is only legally allowed to compensate for certain things, and this happens under the Health of Animals Act, as well as the Compensation for Destroyed Animals and Things regulations. So if you're interested, you can go read those boring legal documents. But essentially, this is about the market value of the animal on the day that they are ordered destroyed. And we recognize that there are often producer concerns around this with the value of animals if they were ordered destroyed in a time when there are not a lot of those type of animals going to market. These animals may not have been for sale, but that is what the law says. CFIA will also compensate for destruction and disposal costs associated with any of those animals ordered destroyed. You can receive advance payments from the CFIA if you request those. Those are usually some portion of an estimated final value, so that cash flow can be recognized in some way with those advance payments. But the process doesn't start until you complete your Form 5945. So if you do not complete that form, that compensation process cannot begin, and it takes longer to settle on a value. There is an appeal process. So within 3 months of receiving your signed compensation form, which I believe is a 4202, you can appeal that compensation, and that is a judicial process. Kind of whatever is ruled upon at the end of that process goes. So there is a process there. If you're not happy with your compensation, you can appeal it. But what about everything else? So we recognize that there is other costs associated with a disease investigation. It might be labor, it might be additional feed, it might be facilities to facilitate testing, or extra help to muster cattle to get those animals in. We recognize that those costs are real and they're experienced by producers, and we recognize that there's a disproportionate reliance on a few producers in a disease investigation to protect the rest of the industry. And with that, we actually have a request for any producers on the call tonight who have been impacted by this tuberculosis investigation. And I will turn it over to Brad Dubois, General Manager with Alberta Beef Producers, to talk a little bit about that request.
Perfect. Thank you, Karin. Thank you, Noel. Really appreciate it. It's been a wonderful presentation, and I hope everybody can appreciate just how complicated this situation is. And I think Karin has done her best to try to walk producers that have been affected, or even just those that are looking to just learn more about it, really well tonight. So appreciated both on your hard work, Karin, and thank you again, Noel, for being here. A couple of things on the compensation piece, though. I want to just say, Karin had mentioned that for any of those individuals that are attending tonight, they can fill out the form, but we will have, this is recorded, and I do believe that we will be able to post it, and it will allow for those that might be affected in the near weeks the opportunity to also fill out this form. So I think, making others aware that might not have been able to attend tonight, really important that we see that. Again, the biggest thing is we really need, are hoping that producers understand that this information will help ABP's efforts. And when it comes to advocating on their behalf, on trying to encourage a program around that additional compensation, or for those herds that haven't been ordered destroyed but are affected by this whole process. We've been working on this and been talking with producers that have been affected since early in 2025, and it's obviously challenging when we're not making the progress that we'd like, but essentially it's all around gathering more information. And we need to help our governments, both provincially and federally, understand the scope of the situation and what producers are going through. And so by completing this form that Karin has got there in the link on this QR code, it will help us in our efforts to try to, again, continue to encourage government to try to find that or put together that program that will aid those producers that have been negatively affected by this. But the biggest thing is that we need producers to understand that it's not a guarantee. It is a lot of hard work on ABP side, working with other organizations to try to help our governments to understand, but it's not a guarantee. But if we don't have the information, we don't have that ability to create that strong argument for why this is important. It also will lead into the potential for other programs, or a program to be developed. Would be what's really important, in my opinion, is that we need a program when events like this take place, for those producers that are on the periphery of this situation, that there is a place for them to go as well. So it's so important. It goes back to those records, keeping track of what's going on, and then helping us to help you. But the biggest message that I'm trying to convey tonight is that it's not a guarantee. We will work our best, our hardest, to try to make something come to fruition. Karin will work closely with Mark Lysing, who's our ABP Government Relations, and we'll continue to flush out what we can to see if we can get to a spot that hopefully provides some compensation, at least recognize the situation of where folks are at. What's super important to also to understand is that we won't be sharing names or any of your contact information. And where possible, we will aggregate the data that you have provided so that we can pull that together and provide that information in a well thought out process to hopefully help in this advocacy process as we navigate this part of the side that's directly connected to the whole TB situation. So I really just want to take the opportunity again to thank everyone for attending tonight. Encourage others to listen to this recording if they get the opportunity. We will do our best through my staff at ABP. They will work extremely hard to see if we can get something across the finish line. But again, it's not a guarantee. But thank you, Karin.
Yeah, thanks, Brad. So there's a QR code there on the screen. You can pull that up, or you can email me for the link. Feel free, if you do email me for the link and folks that have also been affected but don't want to reach out for whatever reason, to pass that along to them. We're hoping that some of this additional information potentially gives us enough information to activate some additional financial supports. But again, as Brad said, there are no guarantees, but we do appreciate any additional information you can provide.